this video we will discuss about insulin secretion in beta cells, that is how the increase in glucose level in the blood vessel will lead to uh, secretion of insulin from the cells. And second thing that we will discuss about is type 2 diabetes mellitus, that is what are the causes and in which case this type 2 diabetes can be reversible and in which case it is not reversible. So let us start with the normal functioning of beta cell or beta cell physiology. So what happens when the glucose level are high in the blood vessels? This will lead to intake of glucose uh, from the blood vessels into the cells with the help of GLUT2, this glucose transporter 2, and this will lead to increase in ATP to ADP ratios as because of the uh, because of the catalytic reaction of this glucose. And with increase in ATP to ADP, it will activates the ATP dependent potassium channel that will cause the intake of potassium within the cytosol of the cells, and this will activate the voltage dependent Ca2 calcium channel, or we can say it will start mobilizing calcium within the cytosol of the cells through different pathway. One of them is voltage dependent calcium channel. Another pathway that will also increase the calcium concentration within the cytosol of the cell is the P2X pathway, P2Y pathway, and this SERCA pathway, that is sarcoendoplasmic reticulum calcium ATPase. All this will cause increase in calcium concentration within the cytosol of the cells, and this happens when the glucose concentration is high in the blood vessels. So, and with the increase in calcium concentration, it will uh, direct the insulin vesicles to move into the extracellular matrix and go into the blood vessels to neutralize the excess glucose that are present at that point of time. So this is how the normal beta cell physiology works. Let us understand what happens in case of dysfunction of these beta cells. In that case, with increase in the uh, glucose level in the blood vessels, glucose will come in the cytosol of the cell with the help of GLUT2. This will cause an increase in glucose concentration within the cytosol of the cells. However, because of this ROS, that is reactive oxygen species, that are more in case of beta cells which are not functional normally, this will prevent the Ca2 plus mobilization and it will promote apoptotic signaling. At the same time, with increase in glucose concentration, the nucleus will generate uh, pro-insulin uh, pro-insulin IAP signals, and at the same time the simultaneously the misfolded IAP and this pro-insulin, since they are not able to form the correct native structure in case of improper beta cells, then this will further exaggerated this ROS effect, and at the same time with the increase in misfolded proteins within the endoplasmic reticulum, it will promote apoptotic signaling. So, and other way that will affect, and the other pathway that will affect the health of the cells, such cells, is the increase in the amount of FFA, that is free fatty acids, or hyperglycemia condition. This will also promote UPR, that is unfolded protein response, and excessive unfolded protein response might promote apoptotic signaling. So all this will finally lead to the programmed cell death of this dysfunctional beta cells. So let us now quickly go through the factors that will cause type 2 diabetes mellitus. So there are some cases that might lead to permanent type 2 diabetes, that is the genetic case, it will cause a decrease in insulin secretion or insulin action whenever the glucose level are high. I have already discussed one video in which I have compared this type 1 and type 2 diabetes, and how these two diabetes are different from each other. So you can go through that video in order to understand this difference between type 1 and type 2 diabetes in detail. In this video we will just understand what are the cases that can be reversible. So in case of genetics it is not reversible, and problem that it might cause is that is decrease in the insulin secretion, or the insulin action is not proper. The insulins are not perform prop efficiently as in the case of normal human beings that does not have these genetic disorders. And the other type 2 diabetes is the modifiable, and this type 2 diabetes occurs because of obesity or low physical activity or unhealthy diet. So what happens in this case, the inflammation is increased, that will cause increase in ROS or reactive oxygen species, that will cause ER, metabolic and oxidative stress, as it is shown here. However, in this case the severity is quite high, whereas in the initial stage when the type 2 diabetes has been uh, observed within this modifiable condition, it is not at that stage, and that can be reversed. So with increase in ROS will leads to increase in this kind of pathways, that is polyol pathway, age receptor activity, hexasamine pathway. All of this will finally lead to increase in this metabolic memory. And these other things are also, this is gut diabetes, inflammation, mitochondrial dysfunction. This out of this mitochondrial dysfunction and gut diabetes, mitochondrial dysfunction occurs in case of genetic disorders, whereas inflammation occurs in case of inflammation occurs in case of this modifiable type 2 diabetes mellitus, and this gut diabetes occurs in can occur in both the cases. So that will this gut diabetes will activate this kind of uh, signaling pathway, it is interleukin 10, interleukin 6, TFN, LPS, pro-inflammatory pathway, long lasting epigenetic changes. And this thing, pro-inflammatory pathway, long-lasting epigenetic changes, are can be caused by gut diabetes or inflammation, and all this will finally lead to increase in this metabolic memory, that will cause either mRNA deregulation, low-grade inflammation, or ROS. And other reasons that can be caused, that cause these metabolic memories are mentioned here, such as with increase in age, the ROS will increase within the human beings, and the respiratory change, or we can say during the respiratory change in mitochondrial DNA damage problems, the ROS can be increased. And this low grade inflammation can also be occurs because of inflammatory response, because of, you can say, some uh, kind of allergic condition, continuous exposure to allergic condition, that might cause inflammatory response, or because of uh, non-continuous exposure to non-self epitope. And this mRNA degradation can occur because of histone methylation, that is mainly occurs in the nucleus where the histone is methylated. And second one is the beta cell mass and insulin secretion are decreases because of the uh, unstable miRNA. So I hope you have understood some basic idea about beta cell physiology or insulin secretion in beta cells, and what are the different kind of type 2 diabetes mellitus, that is which one are modifiable and which one are not modifiable. Thank you for your time. That's all for this video.